Bismillahirrahmanirrahim. Today is our second lecture of genetics. So I will start with a brief summary of the Mendel's laws. The first law says, which is the law of dominance, that some alleles of a gene are dominant and cover up the recessive alleles. And the second law is law of segregation, which says that an organism has two alleles for each gene, but only one of the allele can be passed to the offspring. And the third law says that genes found on separate chromosomes are inherited independently and separately from each other. This is known as law of independent assortment. So here you can see in the diagram, the first law of dominance says that when the parents cross in which one is tall and the other one is short, the offsprings are all tall, phenotypically tall. And when these here you can see the homozygous tall and the homozygous short are crossed, but only the dominant trait, which is tall, is expressed. And the second one is the law of segregation, which says that when the heterozygous tall is crossed with the heterozygous tall, only the dominant gene is expressed more. That is 75% of the offsprings will be tall, however 25% of the offsprings will be short. So the third one is the law of independent assortment, in which you can recall that there was a green rounded pea which was crossed with the wrinkled yellow pea. So here you can see that the two separate genes for separate trait, one for the trait of shape, that is round or wrinkled, and the other one for the trait of color. When they both were crossed together, the offsprings showed separate results. 9 out of 16 were round with green color and 3 were round with yellow color. 3 out of them were wrinkled with green color and only one was wrinkled with yellow color. So the color and the shape were transmitted and expressed separately. So in humans there are some alleles which are dominant, that is for the eye color gene, the dominant is the brown one and the recessive is blue, and the tongue roller is the dominant trait and non-tongue roller is the recessive trait. Brown color is the dominant one, however blonde or light colored hair is a recessive trait. 6 fingers is a dominant trait and 5 fingers is a recessive trait. And there are some more genetic terms like incomplete dominance. When the traits are neither dominant nor recessive and the offspring exhibit the trait which is the blend or mixture of the both parents. For example, if the mother has straight hair and father has curly hair and the offspring gets wavy hair, this is known as incomplete dominance. And then is the codominance. When both of the alleles from mother and father are expressed in the heterozygous state. For example, blood group, when one of the parent is group A and the other one is group B, and the baby which is born can be group AB, so both of the traits are expressed and hence this is known as codominance. And now we come to a few important technologies which are being used in genetics. First is the gene mapping. As you know, the map means the imprint or the direction of a place. So the genetic mapping means the mapping of the genes to specific locations on the chromosome, and gene mapping determines the order of the gene and the relative distance between them in map units. So one map unit is equal to 1 centimorgan, and alleles of two different genes on the same chromosome are known as cis, and allele of two different genes on two different homologous chromosomes are known as trans. And what are the advantages of gene mapping? You can get the better understanding of genetic disorders, and On the basis of this knowledge, you can give the targeted gene therapy. And for the target gene therapy, another technology is very important, which is known as genetic engineering or recombinant technology. What is genetic engineering? It is the manipulation of DNA for practical purposes with the help of restriction enzymes. And what are restriction enzymes? These are the enzymes which cut DNA into fragments that have short, sticky ends. So there are 4 steps of genetic engineering. First is to cut the DNA using restriction enzymes, and second step is production of recombinant DNA, and the third one is to make copies of these genes, and the fourth one is the cells are selected and screened for the gene of interest. So here in the diagram you can see on the number 1 is the DNA is cut. The for example, the human chromosome carrying insulin gene is cut with the action of restriction enzymes. On the other hand, you can see that in the bacterium there is a plasmid DNA where the cut is made, and this human chromosome carrying insulin gene is inserted into the plasmid DNA of the bacterium, and hence the recombinant DNA is produced. This recombinant DNA of human insulin gene is inserted back into the bacterium, where when the bacteria multiplies, this gene also multiplies along with this, and these cells with recombinant human insulin gene DNA are selected and then they are screened. So these can be isolated later on to produce recombinant human insulin. And here you can see the advantages of genetic engineering are manifold. Human genes can be inserted into bacterium for production of human proteins and hormones like insulin, growth hormone, recombinant factor 5, which is a very important clotting factor, and recombinant factor 8 and factor 9 for the treatment of hemophilias. And in genetic engineering, we can also replace the defective genes with the healthy ones, for example, in patients with beta thalassemia. And genetic engineering can also be used to combat bacteria that are antibiotic resistance. So, how will we treat the antibiotic resistance to certain bacteria? We can cut the bacterial plasmid and remove the gene which gives the bacteria power to resist certain antibiotics, so that when that resistant gene is removed from the bacterium, the bacteria is sensitive to the antibiotics. So the plasmids have the ability to replicate in a new location such as in another cell, and by splicing a foreign gene into a plasmid, scientists can transport the gene to a new bacterial cell and replicate it there. As you can see here in the diagram, in the human cell DNA is extracted by using restriction enzyme. This section of human DNA is inserted into the bacterial plasmid gene, and this is the recombinant DNA which further then multiplies, and this multiplication leads to enhanced production of that recombinant gene which can be used later on. So how does bacteria become resistant to antibiotic? As we discussed earlier, bacteria had genes which allow them to survive antibiotics, and they produce and pass these resistant genes to their offsprings. And we also discussed the role of recombinant DNA technology or genetic engineering to combat this resistance. Now, there are certain disadvantages of genetic engineering as well. During the process of cutting and slicing and multiplication of human recombinant gene, you can get many mutations made which can possibly start new illnesses, and it also reduces genetic variation and reduces diversity. This was the lecture for today. Thank you so much, and inshallah next time we'll start the genetic disorders.